Now that we've covered the details of transcription and translation, we need to take a step backward and look at the big picture of what it means to express a gene. In fact, you already know what gene expression is, because if we have a strand of DNA and we go through the process of transcription that you already know very well, and in that process we make messenger RNA, and then that messenger RNA is translated or translation into our protein. If this process is occurring for a gene in DNA, then we would say that that particular gene has been expressed. It's been expressed in the protein. Remember that a cell is not expressing all the genes all the time. They're only taking a particular gene that they need at any given time and going through the process of transcription and translation to actually get the protein. So one of the things we want to ask is, well, how does a cell know, or when is a gene going to be effectively turned on? And then how is it that we could stop this process, or how is it that some genes inside of a cell are turned off, meaning that there may not be transcription or there may not be translation, and ultimately there's not a protein going to be made. So let's summarize here then. When we're thinking about what it means to turn a gene on, means that a gene is expressed and the end result is the protein is produced. However, if a gene is turned off, that means that there's no transcription, no translation, the gene is not expressed, and there's no protein that is made. Now, the answer to this question has effects in many different areas. We want to ask this about cells when we're looking at development, and we'll talk a little bit about that. But we also want to take a look at how this process is controlled. How does a cell in your body right now decide whether a particular gene is going to be expressed and you're going to make a protein in that cell for that gene, and why other genes in that cell are effectively turned off? So I want to talk a little bit about the different levels of gene expression, meaning where are all the places that this gets controlled. You'll notice that there are many levels of gene expression. And so the first level that we're going to look at is what's called the epigenetic level. As you can see, the process of gene expression is controlled at many different levels. So we're going to start at the broadest level called epigenetic level. Now there's a video that you're going to be watching about epigenetics. I'd encourage you to stop and watch that video now. Dr. Neil deGrasse Tyson is going to host that, and I think it will really help you understand what it means to actually control your genes at this level. So I'm going to talk about this with the expectation that you've already watched that video. Remember that epigenetics is talking about above the genome. So epi here means above the genome. And that means that we're looking at whether or not a gene is even available to be transcribed. So we can see two scenarios here in the boxes here that have been blown up a little bit. In this particular example, we can see that our DNA is wrapped very tightly around the histone proteins, and the gene that we're actually looking at right here is this pink gene. And the gene is inaccessible. It means that related to transcription, none of our RNA polymerase could actually get in there. No transcription factors, just chemicals that are going to help enhance transcription, could even have access to that gene. So at the epigenetic level, if our genes are packed and they're not available, then that means that this particular gene is turned off. And while you might have the recipe in this gene to make something, you're not actively making the protein. Let's take a look at a gene that is available to be transcribed and subsequently translated later. And we can see in this picture here that the gene has been unpacked. Those histone proteins have moved away from each other, and now our DNA is accessible, and transcription factors and RNA polymerase could get in there, and we could actually see this gene turned on, and ultimately then potentially a protein even made from that gene. So you can see here at this epigenetic level that there are things that can turn genes on and things that can turn genes off. One One of the things that Dr. Neil deGrasse Tyson talked about in his video was the presence of methyl groups. And we're starting to understand more and more about things like methyl groups, and that when they interact with DNA, they can have a certain effect on them. They can activate or they can repress genes. And then we can have other things that are just termed here epigenetic factors, of course, that can do something similar. They can actually help genes be available and be activated. So remember that when we're looking at anything inside of a cell, it's not only the DNA that you have present in that cell, but it's also whether that DNA is available. So let's actually think back to our cancer case for just a second. Maybe you have a gene that is an oncogene, a gene that could promote cancer. But if that gene is turned off, then it's like you don't have it at all. So these are some ways that we can think about genes. Not only the DNA, the order, the sequence of the nucleotides that you have, but also whether that gene is available. So this is our epigenetic level. Let's talk about gene expression on a couple of other levels. Once the gene has been turned on at the epigenetic level, which we remember would be above the genome, there are a number of other levels where gene expression can be controlled. So let's put epigenetics here. The next level that we want to talk about is what goes on in controlling at the transcriptional level. Remember that for a gene to be transcribed, there are enzymes like RNA polymerase that have to be active. There are also other proteins that are called transcription factors. Transcription factors are proteins, and they are part of showing RNA polymerase exactly where to attach and also to facilitate or enhance transcription. And so if things are working correctly at the transcriptional level, then our gene will be transcribed into our primary transcript of our messenger RNA. But we also know that if there's something wrong at this level, RNA polymerase isn't available or our transcription factors aren't working correctly, then we can also inhibit gene expression at this transcriptional level. Right after transcription, if we remember, we went through an important step called splicing. We also had referred to it as processing. And if you remember, what we did at this step is we had to remove introns and take our exons and put them together. And then we had added a cap and a tail to our messenger RNA before it was ready for export out of the nucleus. There's also some control of gene expression that can happen here, and I wanted to talk in a little bit more detail about what this can look like. At this post-transcriptional level, we know that splicing can actually occur in a number of different ways. Earlier, we just looked at a simple example where we removed introns, we spliced exons together, we put on a cap, and we put on a tail. But now I want us to take a look at all of the different ways that splicing could occur. There are many different examples here, and I just want to focus on two of them. Let's take our first example. If we have our DNA, and we went through the process of transcription and we made this strand right here, which would be our messenger RNA transcript, we can see in gray the introns that are going to be removed, and then we can see in the different colors the exons that we're going to splice together. So when this gets processed or spliced, we actually could take this messenger RNA transcript and splice it in two different ways. You'll notice that the introns get removed, and that if we were to put together our blue and our peach and our purple, we would end up with this messenger RNA. And then that will go on to the cytoplasm at the ribosome to be translated into a protein. And because it contains all of this information, it will make a very specific protein that will fold a certain way and have a certain function. But let's look and see what would happen if when we were splicing, we actually left out an additional exon in addition to the introns, and we only put together our blue and our purple. This would be a different strand of messenger RNA. It's missing some information, but this could go on and be translated and ultimately make a protein that was functional for the cell. However, we know that it's going to be different from this one that's right here. What this means for us is that we could have a gene in DNA, and that depending on how we go through the process of splicing, we could actually end up with 2 different messenger RNAs from it, and then subsequently 2 different functional proteins. Let's take a look at another example. In this example here, what we'll notice is, again, we're going to remove the introns in our final product. But if we, in our first messenger RNA strand here, take our blue and our yellow and our purple, we'll end up with that strand here. If, on the other hand, we take our blue, skip over our yellow, grab our pink, take our purple, then we'd end up with this strand. And again, we can see that these are 2 different messenger RNA strands with different information that are ultimately going to result in 2 different proteins. So it's an interesting question. How many different proteins could you make out of a single DNA? And right now we think that number, in humans anyway, is about 7. Looks like we can take a single strand of DNA, a gene, and actually make 7 different proteins out of it. So this is what we see when we look at gene regulation at the post-transcriptional level. When we're talking about this step right here, we're talking about alternate RNA splicing. And that alternate RNA splicing is just what we saw on the previous slide in the fact that we could make many messenger RNA strands from a single strand of DNA. We also can control gene expression at the post-translational level. So if we move down to assuming now that our protein has been made, here, once our protein is made, we also can influence how this protein will behave. Many proteins have to be activated in order to be functional inside of the cell. So if that protein never gets activated, then it's like the protein isn't even there. It's like the gene never even got expressed and the gene would be off. On the other hand, once this protein is activated and then we have a functional protein inside of the cell, then we know our gene has been turned on and that we have our end result, what we're after, which is that functional protein. So this activation of a protein is all occurring after translation has occurred at what we call that post-translational level. What we have learned about gene expression, the fact that genes can be turned on and off, help us understand a lot more about how it is that organisms can start from a single cell, and then we can have cells of all different types within a single organism. So let's take us for example. We know that all humans start out as an embryo. Early on, these are called embryonic stem cells. We know that in each one of these cells, there's an entire copy of our genetic code in the same way that there is in all of these cells too. So, if a nerve cell, a neuron, has the exact same genetic information as a muscle cell, and that has the exact same genetic information as a pancreatic cell, the question is, well, how is a skeletal muscle cell different from a pancreatic cell from a neuron if they all carry the same information? And it's really about gene expression. There are some of these embryonic stem cells that will go down this path. And what that means is there are certain genes that are going to be turned on, genes for how to become a nerve cell, for example. However, there are other genes that are going to be turned off. Genes about how to be a skin cell or a pancreas cell or a skeletal muscle cell will be turned off in the cells going down this pathway. On the other hand, over here, if one of the embryonic stem cells goes down this route, it is going to turn on genes for how to be a skeletal muscle cell, but it's going to turn off genes for how to be a nerve cell or a skin cell. So you could have cells, these here, that all contain the same genetic information, but because some of them will turn on and off different genes, then they'll all be specialized. This process of turning on and off different genes to become specialized cells is what we refer to as differentiation. Differentiation.